You're listening to Conferences on Lyme Allergy from Children's Mercy Hospitals and Clinics in Kansas City, Missouri. Today is June 13, 2011, and I'm your host, Dr. Jay Portnoy. Our topic today, Molecular Biology of Allergens, Part 2. Our presenter is Dr. Brock Williams. He's affiliated with the section of Allergy, Asthma, and Immunology at Children's Mercy Hospitals and Clinics. So welcome back to Conferences on Lyme Allergy from Children's Mercy Hospitals and Clinics in Kansas City, Missouri. We're joined this morning by Dr. Brock Williams. Dr. Williams is affiliated with the section of allergy here at Children's Mercy Hospital. I believe you're an adjunct professor at the University of Missouri, Kansas City School of Medicine. Dr. Williams is going to continue his series of two on the molecular biology of allergens. He has, in fact, been instrumental in developing methods for measuring IgE to specific allergens and for helping us to interpret them. And although this topic seems to be incredibly complicated, I've been assured that from complexity will eventually come simplicity. I'm looking forward to that unified vision of how all of this fits together. In the meantime, I'm going to go ahead and turn the podium over to Dr. Williams. I'll make your computer the presenter, so you should be able to click the show screen and we'll be able to see your screen. Can you see it?
Mm-hmm.
All right, so take it away, Dr. Williams.
Okay. Okay, how's that, Jay? You got it?
That's perfect.
Okay. Okay, I hope we can get through all this stuff. This is going to be a little more didactic than the last one. The last one was more of the development of the field and over the last 20 years and where all this information is coming from. And now, today, I want to kind of concentrate on the sources of allergens and more or less what they are. This will be somewhat redundant, but you'll find out that that's on purpose, but it's also because you can't avoid that because these allergens are just about everywhere, and there is a lot of redundancy in their sources and where they come from. And really the purpose of this lecture is sort of gain an overall perspective and perhaps give an introduction to the guys that are going to give the clinical stuff, I think, next week. So, let's see. How do I go forward? Okay. We didn't quite get the CME questions from last week, and so I just use these as sort of a review. When you sum up the specific IgE from a large number of tests, like 99 of them, what would you expect the percent specific of the total IgE to be? Well, it turns out it's closer to 20 to 40%, and maybe even closer to 10 to 20% if you're looking at epitopes. But 20% is a pretty good number, and that means there's a lot of cross-reactivity. Let's see. Darn it. Oh, there we go. What percentage of protein families contain allergens? Only about 2%. So allergens are special structures in nature. The common properties of allergens include the following, except heat stability, stable to digestion, tend to form polymers, infrequent in nature, involved in protection. And the answer there, of course, is they're not infrequent in nature. On the fourth question I gave, the common functions of allergens include the following, except proteases, protease inhibitors, lipid transport receptors, and calcium modulators. And the answer there is receptors. Okay. So we've kind of used those questions to kind of review the fact that only 2% of all the protein families, which there's about 11,000 of them, actually contain allergens. And the cross-reactivity is very common among these different allergens, and the cross-react is exists between similar and diverse sources. So you'll see that quite clearly in a few minutes. Sensitization by proxy might be common, like we were talking about tropomyosin J. Not every member of a family is allergenic, and not all members of a family are cross-reactive, but many are. And that's kind of where the rub comes, because we know it's the same protein. It comes from 2 different species of plants, but it may not be cross-reactive, but most of them are. So, clinical reactivity is somehow related to structure, function, and the route of sensitization. We'll discuss that somewhat. So the general properties, they're small to medium proteins, mostly stable to proteolysis. They're thermostable, and the reason they are is because they have multiple disulfide bonds, which kind of hold them together. They do have a propensity to aggregate. I'll show you a slide, I think, in a minute here. They're very pervasive in nature and involved in very important, somewhat universal cellular and tissue functions. And here's this slide is probably difficult for most people to see, but if you concentrate on DERP2, for example, that when you get a dimer, all these proteins, these are major allergens, and they all tend to aggregate. But when they get a dimer, look at the difference in the structure. Look how different this is from this. And usually the allergen epitopes are exposed loops on these. So you can see that this is good for 2 reasons. This makes this allergen nice to cross-link for IgE, and that it's different from the primary structures that we usually draw. So these things can change upon aggregation. The general functions that we defined include of the allergens that include lipid metabolism, the lipocalins and lipid transfer proteins, calcium regulation, parvalbumins in animals and full calciums in plants. They're involved in motility and signaling, the profilins and the tropomyosins. Defense, we'll talk about pathogenesis-related proteins, and also the trypsin alpha amylase inhibitors, lipid transfer protein storage proteins, that's the peanut allergen, the RH2, and it's in a lot of different sources. And there are glycosylases that seem to be very important. There are proteases that are also common in venoms, but they're also in almost all organisms, and we kind of look at these as disturbing some of the functions of the proteases and anti-proteases that we have in our own systems. And there are a group of allergens called expansions, which are involved in grass allergy mainly, and they're involved in the growth of plants to allow them to expand. Okay, what are the typical animal food allergens? This is the top 5 by ingestion. The structure of the proteins, the EF hand proteins, which is a description of their structure, are calcium binding proteins. The tropomyosins are involved in motility, not on just of cells, but muscles and this sort of thing. The alpha and beta caseins are rather important. Transferrins seem to be important, and they're also important in our defense against microorganisms, in that they bind iron and sequester it from bugs. The lipocalins seem to be very important, and they're simply fatty acid binding and transport proteins. These 5 top families account for 66% of all the plant animal food allergens, and of which there are 95 of these that have been defined in 144 different out of 144 in 26 different protein families. An interesting study is Jenkins' evolutionary distance from human homologs reflects the allergenicity of animal food proteins. And what it shows is that if it's less than 62% similarity or homology with the human protein of the same kind, then it can become an allergen. And this is a very interesting study, and since we're animals, we have all these proteins too. So the ones that differ by over 62% are the ones, or less than 62% similarity can be allergens.
So the further away on the phylogenetic tree they are, the more likely they are to cause a reaction.
Well, yeah, that's true. That's actually true, but there are some exceptions, and it kind of depends on where those differences are in the protein. If they're on the surface, then it's probably relevant. If they're interior, they're probably not. Okay, what are the major fish allergen proteins? There's little doubt about this one. It's the beta-parvalbumin. It's identified in a lot of different fish. It's an EF-HAN domain signaling calcium binding protein. It's involved in transport and signaling and important functions. And you see it in all these different fish, all the way down the line. But you also see it in shrimp. You see it in lobster. You see it in frog. You see it in crawfish. And man even has his own alpha and beta parvalbumin. So the interesting thing there is that the alpha parvalbumins have not changed very much during evolution, but the beta parvalbumins have, and that's probably why they're more potent allergens. As a matter of fact, this is a really good-looking fisherman. This is a small pike that I caught up there, and pike is really kind of interesting because its beta parvalbumin is very close to humans. It's over 62% homology, and therefore doesn't fall in line with the rest of the fish. So that's just a good example of how these things can vary. Now, beta parvalbumins in fish and amphibians, but the polcalcins are sort of a similar function, but they're in plants. But there are these EF-HAN domain proteins that bind calcium, including troponin C, calmodulin. Calmodulin's definitely involved in all the G protein coupled receptors. And these things are, in general, muscle contraction, signaling, calcium binding, transport, and calcium influx is really one of the things that we recognize as turning on a lot of functions in cells. Sort of give you an example of these EF-HAN domain proteins, the parvalbumins in fish and animals, and then the polcalcins in plants. So these are all very similar proteins. They can become allergens. And of interest here is the parvalbumins, BLA-G6 and 8, are actually from cockroach. And BOS-D3 is a calcium-binding protein from milk, which also cross-reacts with these parvalbumins. So you begin to see that we're jumping across different species lines, and that these things can be common in nature. Okay, seafood allergy, and we should have thrown a couple cockroaches and maybe a few insects in here crawling around because they're very similar. And so it's something that you have to think about again with this process.
Sure, Red Lobster. Not only that, but if you find a cockroach on that plate, you can get your meal for free.
It depends on which country you're in, I imagine. At any rate, tropomycins are a helical coiled coil, and they have 7 actin binding sites per unit. So that makes them nice to cross-link IgE on the surface of inflammatory cells. And they're very stable. They're very highly conserved. And one thing of interest, even with this high conservation, the vertebrate tropomycins are very quickly digested by pepsin. So they're a good food source. but the invertebrate ones are not. And invertebrate ones include the insect, moth, fly, spider, midge, mite, silverfish. I mean, you can name them all. And the foods, of course, are shrimp, lobster, crab, abalone, snail, mussel, octopus, squid, and oyster. Even in parasites of fish, the Anisakis and of course, Ascaris. So you see that the tropomycins are, obviously quite common in nature, but also we have to realize that perhaps the tropomycins of all these insects don't have absolute cross-reactivity with the foods, and some of the foods, for example, don't have absolute cross-reactivity with each other. For example, shrimp and abalone. So these things are still working out, but it's pretty well established that the degree of reactivity is related to the degree of similarity between any two sources of tropomycin. Okay, what about milk allergy? It's kind of interesting. I put some just notes up here so I wouldn't forget about it. We all know about lactase insufficiency, but one interesting note is that casein, limiting digestive casein, releases an opiate, which slows down the motility of the gut, and therefore the stuff hangs around longer and perhaps gives you longer time to become sensitized to these proteins. Another interesting point is that TGF-beta is in fairly high amounts in mother's milk, and we know that really helps to establish T regulatory cells in the gut. The question I would ask, and I don't know if anyone's answered this, is that when we pasteurize milk, do we destroy this TGF-beta? So, interesting thing to consider. At any rate, major milk allergen proteins include lipocalin, which is, again, a fatty acid carrying protein, calcium binding protein, which we've seen before. BOSD4 is alpha-lactalbumin, which is a C-type lysozyme. And we'll see this all over the place, too. And we see it in egg, in GALD4, and it's a pathogenesis-related 8 protein, which we'll discuss these in a little while, from plants. So alpha-lactalbumin has a number of different sources from which individuals can become sensitized. BOSD5 is beta-lactoglobulin. It's another lipocalin, and it actually cross-reacts with dog, KNF1, 2, and 6, cat, FeLD4 and 7, horse 1 and 2, and cockroach, BLA-G4. So you can see some relationships here also in across species lines. BOSD6 and 7 are BSA and the albumin and the bovine IgG, and they're minor allergens. And of course we have the caseins in BOSD8. So when we talk about lipocalins, I think they're really important all by themselves because they're one of the major sources of allergy to animals. They essentially transport small hydrophobic molecules, steroids, retinoids, and lysophospholipids, but the retinoids is kind of interesting because we know that retinoic acid is another one of these factors which in the gut stimulates T regulatory cells. And so if you got something that binds it, then maybe you don't get that stimulation as much as you should, and perhaps that allows you to make more aggressive immune responses to these things. We know that the lipocalins are involved in cell adhesion, motility, cytoskeleton changes, proliferation, survival, prostaglandin synthesis, membrane biogenesis. They carry pheromones in animals, and they're involved in G protein signaling. They're all over the place: bacteria, vertebrates, invertebrates, and plants. So the important lipocalin allergens, we already mentioned a few of these, but from the dog, KNF1, 2, 4, 6, cat, FeLD4 and 7, horse, cow, mouse, this is a cockroach, Perry4 and Blage4 cockroach, rats, and even a mite, Duraf13, is a lipochelin. We also know that man have a tier lipoprotein, lipochelin, which also can be an autoantigen. Ticks have a histamine binding protein, which is a lipochelin. And we know that it's in renal injury, the neutrophil gelatinase-like lipoprotein is a measurement of severe renal damage in humans. So it's all over the place too. And it's involved in defense mechanism, chemical and metabolic signaling, and it's widespread. Okay, here's a ooh-ah picture. Right. But, and here's the rest of the story. The cats have 8 identified allergens, and the uteroglobulin is FeLD1, and that's possibly a lipochelin also. I just couldn't find any information on that. Cat albumin is FeLD2. Cystatin, FeLD3, which is a cysteine protease inhibitor. FeLD4 is another lipochelin. 5 and 6 are immunoglobulins, which are minor allergens, but can be potent. FeLD7 is a lipochelin that resembles von Ebner gland protein. And FeLD8 is one I wasn't too familiar with. It's called latharin. It's in their sweat, but it's really responsible for distributing oils into their hair, and so it gives them that shiny look and healthy hair. And dogs, CanF1, 2, 4, and 6 are all lipochelins. And by the way, CanF2 and FeLD4 are highly cross-reactive. So it's sensitive to the cat and the dog at the same time if you're sensitized to this particular lipochelin. But CanF5 is a trypsin and another protease, which we see in a lot of other sources as an allergen. So, what about egg? Okay, egg, we've already talked about this a little earlier this morning, but Galdi is ovomucoid. It's present. It's 11% of the protein of an egg, and it's a serine protease inhibitor with 3 similar domains, which makes again nice for cross-linking. It's also when humans are lacking this in their pancreas, obviously the trypsin can start digesting the pancreas, and it's responsible for pancreatitis in people who have mutations of this particular protein. It's also a pathogenesis-related 6 target of mold. Molds make a lot of proteases to digest the environment, and for plants to keep them from digesting them, then they secrete a similar serine protease inhibitor that actually inactivates the protease. Galdi-2 is ovalbumin. It's also a serine protease inhibitor, but it's thermolabile, and it's actually 54% of the protein in an egg is ovalbumin. So there's a lot of it, but it gets when you cook it, it gets denatured and therefore doesn't cause so many problems upon ingestion. Galdi-3 is a conalbumin, a transferrin, another one of these proteins that carries iron, and again, it's active as an antibacterial. And C-type lysozyme, which you've already mentioned before, which is a beta-1,4 acetylmuramidase. This is a very interesting protein. Actually, the guy who discovered this is Fleming. He's the same guy that discovered penicillin by accident, and he discovered this one by accident. So the guy was accident prone, but took advantage of that. What he did is he dropped some raw egg, and I don't know how the heck he did this, on one of his bacterial plates, and noticed that the bacteria were getting lysed, just like penicillin inhibited the penicillin mold inhibited the other bacteria.
So bad lab technique is a good thing, huh?
Yeah. So well, if you pay attention to try to figure out how you messed up. And what caused it. But also, we mentioned this before with BOS D4 in milk. There's this E-type lysozyme. So you can really see that you might have a cross-reactivity between egg and milk. Vitagelin is another protein. It's a lipid transfer protein. We've mentioned these before, and they seem to be important. They bind to estrogens, and they're also common in cockroach and mite. So vitagelin's kind of all over the place too. So here's the food allergens that we've discussed: the calcium binding proteins, the EF-HAN proteins, milk, the BOS D3 calcium binding protein, and the parvalbumins from fish, tropomyosin, shellfish, insects, along with tropin and myosin are also allergens from these sources. The lipocalins, animals, insects, they carry lipids. Protease inhibitors, egg, and glycosylases, the lysozyme. So these are the major groups of animal food allergens that we're dealing with. Okay, let's get in a few others. Peanuts. We think peanuts is such a culprit, and we see that it does have quite a few important allergens. But I'll point out that ARA-H1, ARA-H1 is kind of interesting. It's bacillin. It's a trimer. It binds chitin. And we're going to find this in a lot of plant allergens, that they bind chitin or they're chitinases, such as latex. They're involved in storage and protection, and it actually binds insulin. It has been used to treat hyperglycemia. It's recommended. You can look it up on the internet, and it's kind of interesting. ARA-H2 is congluten, but also ARA-H6 is a congluten from the same family, and they're both amylase protease inhibitor seed storage lipid transfer proteins. In other words, they have multiple functions besides just being a source of nutrients for the new-growing plant. But amylase is really an important inhibitor in plants, and the reason it is because fungi, insects try to, especially the larvae, need a really good source of glucose. So they infect plants, and they start chewing this up with their amylase, but the plants have an amylase inhibitor that prevents that, and the larvae insects die because they can't get enough sugar. So it's pretty good. It's pretty good protection. ARA-H1, 3, and 4 are very similar proteins, but this glycinin, which is sort of like bacillin, forms this a homohexamer, very large, and it hangs around the gut for a very long period of time because of this. We have propylins, which we'll talk about in a minute, in all plants, and ARA-H7 is another congluten that not much information on, but ARA-H8 is a PR10 protein, which is very important in a lot of plants, but as an allergen, but it's also somewhat heat labile, and therefore probably is responsible in many instances for oral allergy symptoms as opposed to systemic symptoms, because it breaks down. That probably depends on the dose and how much you ingested of it. But it's an RNase, and it's there for the plants to protect themselves from viral. The ARA-H9 is a nonspecific lipid transfer protein, which we've obviously mentioned these before. And RH1011 are olefins for oil storage, which kind of keeps the oil in little packets, but haven't really, I don't think they're very dominant allergens. So there's friends of peanuts allergens, and these include the vicillins, which we pointed out was RH1, 3, and 4. They're also present in amaranth, sesame seed, pea, bean, cocoa, walnut, lentil, hazelnut, and cashew. So, obviously, if you're sensitized to these or you have a positive test, it could be from any of these sources. Same thing goes with conglutens. They're RH2, 6, and 7. They're also present in almonds, rye, wheat, soy, and peanut. So we have to be cognizant that there's a lot of different sources of these things out there, and that, you know, obviously cross reactions are possible.
So an IgE to peanut could be due to an IgE to one of these cross-reacting proteins and stuff?
Exactly, exactly. And it probably, the clinical reactivity probably depends on the extent of that cross-reactivity. So you might be measuring this stuff, and it may not be cross-linking, or, you know, vice versa. So those are things that, you know, if we look at the amino acid sequence or the tertiary structures and we see that they're very, very similar, then yeah, they'll react to both. If they have the IgE. Okay, what about the wheat grains? Tri-A2, we don't know. I haven't seen defined as any kind of function. But Tri-A3 is really a pollen allergen, and it's one of these expansins. The expansins allow the plants to grow, as I said earlier. Propylene, Tri-A12, is present in wheat, but there are 4, Tri-A14, 28, 29, 30, are lipid transfer proteins. And this just keeps popping up all over the place as an allergen. Tri-A18 is a chitin-binding protein, very similar to heavin, the major allergen of latex, HB6. And Tri-A19 is kind of interesting. That's your omega-gliadin, and that's actually an amylase protease seed storage lipo transfer protein. So here it is again. There's thioreductases in wheat have been identified as allergens, and the glutenin, which is responsible for the viscosity of bread, is also an allergen. Also, it's kind of interesting, the PR5 pathogenesis-related 5 proteins, the thaumatins, which are very sweet to the taste, are also very common in all the grains, and we'll discuss that in a minute when we discuss pathogenesis-related proteins.
Are any of these associated with celiac disease?
Oh yeah, gliadin is, and essentially it's what is it? The deaminase. This is a very basic protein, and when it's deaminated, then it becomes a potent allergen that stimulates some of the, you know, the cross-reactivity with it. So, you know, you get gut problems from it, right?
Mm-hmm.
Yeah, I mean, that's yeah.
So the Tri-A19 is a big player there.
Yeah, that's a big player. Okay. Okay, here's our friend the dust mite. And we know that it contains a number of very important allergens, and these include the DURF1, which is a cysteine protease. But we also know cysteine proteases are present in all these fruits: papaya, bison, kiwi, pineapple. So that could be relevant. DURF3, 6, and 9 are serine proteases, similar to our own thrombin. They're present in cockroaches and venoms, and that KNF5 dog antigen I mentioned. 15 and 18 are chitinases, and that's similar to the pathogenesis-related proteins 3, 4, and hevine. But chitin is really an interesting molecule. Actually, what's really interesting is the chitin fragments. whenever you chew up chitin, you get these fragments, and they're highly immunomodulatory in favor of Th2 responses. So that's probably relevant to all the chitinases. A lipid transfer protein, PR14, DURF2, 7, and 13. We've seen this before. The tropomyosin, we talked about that with shellfish and insects. Myosin tail is associated with the tropomyosin. And alpha amylase, the enzyme that goes in to digest the sugar or the starch. And we've seen this in relation to the cereal alpha amylases, and this is an important allergen in Baker's asthma. We see the glutathione transferase, which is also seems to be important in cockroaches and also fungi. A lipoprotein, which is similar to vitellogenin in egg. That's the one that binds estrogen. And DURF5 and 21 might be exclusive to mites, and we don't know what their function is yet. They're pretty similar to each other. Ah, there's a good one.
There you go.
Right before lunch, there's yesterday. It's probably about 3 days, 4 days. But we should say a few things about molds. These are the major allergens from molds, but there's quite a few others that are identified as allergens, but not necessarily as prevalent. The serine proteases, the molds secrete these to digest things in the environment, proteins, so they can absorb the amino acids. But it's also, serine protease is a major allergen of muskmelon. The enolases seem to be very prevalent mold allergens, and I can't really explain that one. It's the enzyme, if you recall, the glycolytic pathway, where 2-phosphoglyceraldehyde or glycerin is made into phospholinopyruvate, which is a very important step because the next step you get some ATP out of. So, but the enolases seem to be very important, and it seems to be a highly cross-reactive allergen from a number of different molds. There's a 6DS ribosomal protein, which seems to be a very common mold allergen, and I don't think anyone understands how that one works. There's the thioredoxins, the redoxin, and that I mentioned earlier. There's catalase, of course. Molds are inhibited by peroxide, and a lot of plants can make peroxide, and one way to avoid that influence is to produce catalase, which destroys the peroxide. There's the spartyl proteases in molds, which is also present in BLA-G2 and also in the conglutinins from peanuts. Glutathione synthetase is from molds. It's also a cross-reacting allergen, and it's also present as DURP8 in mite and BLA-G5 in cockroach. And there are even heat shock protein 70 homologs, which are also present. They're also present in hazelnut, but they've been identified as major mold allergens. So you kind of see nature kind of getting together on all these sort of similar functions and similar protein structures from a lot of different sources. Let's see. I was going to say something about this, but this is we're talking about grass and plant allergens. But most people look at this and say, "Oh my gosh, that's grass." But actually, the grass that you're allergic to is the pollen mostly, and that's why we mow our lawns so we don't get some pollen. But if we talk about all the plant allergens together, we talk about plant food allergens and plant inhalant allergens, and the line between these is somewhat blurry, because you might be sensitized as a food allergen to some plant inhalant allergen, or vice versa. So we don't know. I think we have figured out a few cases, but you That's still up in the air, but the possibilities have to be recognized. So we have these alpha amylase trypsin inhibitor lipotransfer proteins, the prolamins, which we've talked about, and they're very broadly distributed and important allergens. The cupins, as the bacillins in peanuts, the 7-11S storage proteins. Propylins are very important allergens, and I think it accounts for about 20% of the sensitization to plants. The BET-V1 analogs, which are PR10 proteins, are very prevalent. Thaumatins, PR5 proteins, very prevalent. And the chitinases, again, the PR3, 4, 8, 11 pathogenesis-related proteins. This accounts for 67% of all the plant food allergens. I remember I was going to say about that golf course. I'll come back to that in a minute. Plant inhalant allergens include these expansins, which allow plants to grow and still maintain their cell membranes, their cell walls. Of course, the propylins are also plant inhalant allergens. These prolaminins, the alpha amylase trypsin inhibitors, lipid transfer proteins, are also EF-hand, calcium binding proteins, the polcalcins, which we talked about before, and the BET-V1, the GRAS 5 and 6 allergens, which are again PR10 proteins. Pectate lysases seem to be more important as inhalant allergens than they do as food allergens. But these groups right here account for 60% of all the plant inhalant allergens, and they're widely distributed again. The expansives are they're really interesting. They're in all sorts of plants, but they're mostly allergenic from grasses. 90% of the grass allergics have IgE expansins. They're very stable, and they're involved in the cell wall expansion. They kind of loosen up the carbohydrate chain so that things can expand in a really neat mechanism, but we won't go into that. They're involved in pollen tube opening, and they're very similar to the chitin binding domains of several chitinases. And of course, they're present in all these grasses, but they're also present in fruits like kiwi, grape, and potato, which is not a fruit. Okay, a few words about plant pathogenesis-related proteins. They're widely distributed in the plant kingdom. They're really the plant innate immune system. 23% of all plant allergens are in this family, so they're very important. They have orthologs or homologs in yeast, insects, and vertebrates, which we can consider as a cross-sensitizer. They're remarkably protease resistant. They're malleable. They provide protection mainly to fungi, but also insects, nematodes, viruses, and herbivores. They're just the immune system of plants. They're mostly they can be constitutively produced or in specific adverse events that happen to plants, whether it's weather, chemical damage, they produce more of these. The ones underlined are the main allergens here. These are all the PR1 pathogenesis-related proteins. There's 15 to 16 of them defined. But the main ones, again, are these the beta-1,3 glucanase, the chitinases, 3 and 4, and thaumatin, which this is, thaumatin actually, once a plant can kind of digest the cover off of a mold, then thaumatin is an osmotin which can puncture its membrane and its cell membrane and allow leakage and do damage to the mold. They're also, I think, somewhere like 10,000 times sweeter to the taste as a protein than sucrose. And in Europe and several other countries, they use it as a sweetener. So, kind of interesting sideline. I don't know how much allergy that causes, but nevertheless, they use it. And the PR10...
allergy syndrome as a result.
The what?
I was wondering if it causes a lot of oral allergy syndrome as a result of that.
You know, I really don't know. I haven't heard. Maybe they're tolerant to it because they eat so much of it.
They desensitize, yeah, there you go.
Yeah. And again, the lipid transfer proteins, PR14, and the RNase, the PR10, which is the BET-V1 homolog. These are really important proteins. And as sort of a sidecar here, you know, we talk about the hygiene hypothesis as being responsible for an increase in allergy in modern societies. It essentially says that our innate immune responses modulate our adaptive responses, and if we're too clean, we're not stimulating these enough, and so we're getting aberrant responses. But I think we got to stop and consider this might be more linked to the plants than the humans, and what we've done. These responses to pathogens of plants cause an increased plant pathogenesis-related proteins, all of them. And so it could be our growth methods that we're actually using to produce our foods that increase the number of these, the amount of these allergens. And in particular, when we ripen things like tomato with ethylene gas for 2 days because we pick them green and then we want them to be nice and red, they don't taste anywhere near as good as vine ripened, but they have a whole lot more pathogenesis-related proteins in them because ethylene is a plant hormone. So it could be possible that the increase in allergy in modern societies might be linked to our production methods. And by the way, most of the things they're trying to do to improve our food is to make it insect resistant, and these pathogenesis-related proteins are the ones that they're trying to put into or increase their production in most plants to keep them more insect and mold resistant. So, possibility. Okay, I'll get a few minutes here and I'll talk about profilins. They're present in all eukaryotic organisms. They're very highly conserved, and almost 75% identity among distant related organisms. And profilins bind actin monomers and keep it from polymerizing. And so they're involved in cell locomotion, even in mRNA splicing and membrane trafficking. And they're really kind of important little proteins because they bind to phospholinositol pyrophosphate, and they're released by phospholipase C, which we know releases these second messengers, which activate a whole host of other things, the diacetylglycerol and the inositol triphosphate, which causes calcium stores to be released. And so these are very much involved in a lot of cellular activities, and profilins actually are involved in those activities also. 10 to 20% of the pollen-sensitized people are sensitized to profilins. They're important in vegetable allergies, respiratory allergies, and they might be really important depending on their dose and also their ability to create bystander effects. In other words, if you're sensitized to a profilin and you create an IG milieu in a particular tissue, then it's likely to stimulate IG production to other things that are accompanying that particular source of profilin, which are a lot of the other allergens. Okay, just a few words about the seed storage trypsin alpha amylase inhibitor lipotransfer proteins. This is the protein family 234. They shuffle phospholipids, fatty acids. They're very stable, and they're remarkably similar in all higher plants. They're responsible for a lot of the cross-reactivity between fruits and vegetables, and there are multiple genes in most plants, and therefore some of these might be more potent than others as far as allergens, and that hasn't been defined yet. kind of interesting sideline is the foam on beer is due to lipid transfer proteins.
Hmm. Good stuff, huh?
Yeah, it looked good to me. So kind of reviewing stuff, we got expansins and grasses, shellfish, tropomyocyns, fish parvalbumins, animals, the lipocalins, and the plant foods, and also some of the pollens, propylins, lipid transfer protein, osmotins, amylase protease inhibitors, glycolases, and chitinases, and the thaumatins. And that's it. There, that's simple, isn't it, Jay?
Oh, wow. Out of complexity comes simplicity.
Yeah, right. Let's see. I don't think I have to review this, but maybe I'll say a few words about mechanisms real quick. I remember the comment I was going to make on the grass, on the golf thing, was that grasses and grass pollens not only come with the allergens, the expansins, and this sort of thing, they also come with phytoprostates, which are essentially sort of like a plant prostaglandin, and they actually stimulate Th2 responses. So there's another, the allergens don't come by themselves, and they can actually, that actually might be an explanation why so many people are allergic to grass. Proteases actually increase permeability, they digest surfactant proteins and tight junctions, so allergens can get in better. They have some very direct immunological effects. They digest CD40, CD25, CD23, and they had protein-activated receptor 2. These are all very important. CD40, you can't get a Th1 without CD40.
Mm-hmm.
And CD25 is the alpha chain, I think, of the IL-2 receptor, which is also, involved in Th1 and Th2 responses. The CD23 is the low affinity receptor for IgE, and it seems that the soluble form stimulates IgE production. And of course, the PAR2, the protein-activated receptors, are very important in hormone effects. And if you come along and activate that with a protease, then obviously you're going to be activating some functions in cells that they prematurely or overly